food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode.
Yes, so currently I am MD PhD and I'm both a practicing physician. I am a dermatopathologist. So this high level of specialization in pathology. So I read the slides in skin sent by dermatologists, and I provide the diagnosis, which is guiding the dermatologist or surgeon or anybody else with therapy or excision. So this is one. On the second, I am also a scientist. I'm PhD. I got my PhD in Poland in tumor biology, but my areas are diverse: biochemistry, molecular biology, cell biology, skin biology, and neuroendocrinology and photobiology. So these are these areas. My current appointment, I am an endowed professor of dermatology at the University of Alabama in Birmingham. I'm also a professor of pathology here. And I have second appointment, 5/8 appointment of the Veteran Administration here in the Birmingham, where I predominantly serve as a dermatopathologist. So I do the clinical work, but also I'm supported by because there is another benefit I can apply through the Veteran Administration system for the grants, and I'm supported by the VA Merits Awards, which was renewed for another 4 years after peer review. And at the University of Alabama, I am supported by Department of Defense grants and by the grants of NIH and matching internal funding. It is simple. If you are at the UAB, depending on the department, you're successful with NIH or other foundation, they will support you even more, so you will be more successful.
So you're an MD PhD, so you do both clinical work and you do research, and a lot of it is focused on different aspects of skin biology.
Yes, this is correct.
One thing that I know that you've studied is you've studied the function of hormones and the endocrinological aspects of skin function. Normally, when we think about endocrinology and hormone biology, we think of endocrine glands like the pituitary gland or the adrenal glands or thyroid. But to what extent is the skin an endocrine gland? To what extent are hormones being produced within the skin itself?
So I wouldn't call it endocrine gland, because endocrine gland, you have the gland which is highly specialized and only it's like anterior pituitary produce several hormones like adrenals. You have adrenal cortex producing glucocorticoids or any other classical endocrine gland. The skin is organ with a neuroendocrine capability. So depending on the environmental stress and the context, it's not only stress, but also is sensing the environment, because this is the most exposed, outer exposed part of our body, but the part of the body of different animals through which they communicate with the external environment. So this is how so the external environment is not only recepted by other senses like eyes, hearing, smell, but also through the skin.
Mhm.
Just so the skin can sense the temperature, can sense the light, can sense different wavelengths of the ultraviolet light, and also be subject to different stressors. So in reaction to the stressors, depending on the stress applied and depending on the anatomical location in the human skin, it can start to produce several classical neurohormones. And in fact, I published in 2000 in the Endocrine Review, which is a leading journal in endocrinology, with Dr. Wartzman, a review neuroendocrinology of the skin, so which was for the endocrine community, it was a little bit shock that the skin, where we were summarizing our work of other people, that the skin can produce classical hormones that are produced by the endocrine glands or by the brain, like corticotropin releasing hormone or corticotropin releasing factors, because they use people, they say CRF or CRH, depending who say, and in which context they can produce corticotropin releasing hormone related peptides such as urocortin 1, urocortin 2. They produce the classical, and this is prohormone like propiomelanocortin, which is further in the regulated way processed to ACTH and further down to alpha-MSH in the context-dependent version, or beta endorphin. So you have opioids, and this can act locally, but under the significant stress can be released into systemic circulation. But under, as I said, under highly significant stress, and then have the global homeostatic effect. Another hormones which are produced in the skin is hormone of happiness, serotonin. So serotonin is produced in the brain. You are a neuroscientist, so you know it is produced in the GI, but the skin is another organ which is producing serotonin. And the serotonin also in the skin can be biochemically transferred into melatonin, another. So this is a site also of another very important hormone with that also context-dependent different functions are of melatonin. There are also several other hormones. There are glucocorticoids. In the human skin, cortisol is produced in the skin, and there is hormone metabolic pathway for this production, which is inducible, because all of this production is inducible. On the low level, when it will be very low. It will be almost below detection, but depending on the stressor, depending on the signal, it is inducible. So cortisol, corticosterone in rodents. And then we have the expression of corresponding receptors in the epidermis and different dermal structures. So these hormones will act locally to regulate the local homeostasis or to have phenotypic effect in the context-dependent fashion. But if the stress is huge, it can be released into the circulation and have the systemic effect. And Another example, it is a little bit immunology, but different cytokines, interleukin-1, interleukin-6, TNF-alpha, are also produced not only by the immune cells, but produced by skin cells. And the skin cells, like keratinocytes or fibroblasts, are immunocompetent cells. And these are classical immune signals. But they have also consequences in the signaling on the neuroendocrine level, because the cytokines interleukin-1, 6, they can stimulate also propimelanocortin expression and processing, and stimulate corticotropin releasing hormones. So they can activate also as another type of the signal, the hypothalamic pituitary adrenal axis. And maybe the most classical hormone, not hormone, is prohormone. So this is a production of vitamin D, where it is only the skin or any other side of the body which is exposed to the ultraviolet B radiation, where the 7-dehydrocholesterol, which has the unsaturated B ring, two double bonds on the B ring, and this is crucial. It's phenomenal in the evolution of life, how the life adapted the structure for the signal transduction after absorption of ultraviolet B, not ultraviolet A.
Yeah.
Not any other. Only ultraviolet B is being broken, and then through the photochemical and thermally driven reaction change in the vitamin D3, which is injected into the circulation, and then after activation by the two classical enzymes, is acting as an active form of vitamin D, having the enormous homeostatic activity, not only on the bones or skeletal muscular system, but also several other regulatory functions. In the classical way, it's acting through vitamin D receptors. And in fact, I don't know if there is any organ that does not express vitamin D receptors. So this is classical pathway. I covered also non-classical, alternative pathway, which is activated by classical enzyme of steroidogenesis type 11A1, and which hydroxylate vitamin D in different positions, producing different active forms that are also subject of modification by other cytochrome enzymes. So we know at least that right now there is at least 18 additional hydroxy forms of vitamin D, in addition to the classical described in the classical pathway, and many of them are circulating in the human body, and also murine. We didn't publish about the murine, but we know for years, because we use some as a controls experiments, and they have similar effect like classical carcitriol, but also sometimes different, and also similar but different mechanism of action, because not only vitamin D receptor is the target for the regulation, but also several other receptors which we have discovered. It's like LXR, which is liver X receptor, like aryl hydrocarbon receptor. These are example of the receptor, or retinoic orphan acid receptor alpha and gamma. With this one, they act as inverse agonists. You want to?
Yeah. So there's a lot to unpack there. Basically, it sounds like the skin is not an endocrine organ like an endocrine gland. It's not fully specialized to only specifically produce and secrete hormones, but nonetheless, it does have the ability to produce and secrete and respond to various hormones and related signaling molecules. And it sounds like a lot of that has to do with the fact that because the skin is our interface with the environment, it is going to encounter many different forms of potential stress, physical stress, changes in light, temperature, and so forth, pathogens that might get in through the skin barrier. And because it has to deal with all those stressors, that is part of the reason for why it can actually produce certain hormones.
This is correct. And and it has certain pathways like primordial in the primordial form, which are embedded into the skin, which we have this organization also on the central level. I'm giving the best example on the hypothalamic pituitary adrenal axis, which is a really very beautiful axis and directing the responses of our body, perfect responses, perfect organization to stress, and which really followed the concept of Hans Sely and then others, Willie Vale. So you have hypothalamus producing the corticotropin, and you will appreciate this as a neuroscientist. Hypothalamus producing corticotropin releasing hormone or factor, and then which enter portal circulation, go to the anterior pituitary, act on the melanocortin receptor type 2 in the anterior, in the corticotroph, stimulate release of ACTH, and also stimulate the PMC production and processing to ACTH because it has to be replenished. And the ACTH in the circulation is going to, is a classical adrenal cortex and acting, no, in the anterior pituitary, we have corticotropin releasing hormone receptor type 1. With the MC2, I went too fast. Type 1, which after activation, release of ACTH circulation, adrenal cortex, action on the melanocortin receptor type 2, which is a very specific for ACTH, recognized predominantly and almost only ACTH, and then release of glucocorticoids in humans, cortisol and different related species. In rodents, majority corticosterols, they do not produce cortisol. And just counteraction against the stress and counteraction against the immunosuppressive immunosuppressive effect and modification of the global body homeostasis. And this beautiful feedback mechanism. Cortisol is stopping production of CRH, is stopping the PMC and production of ACTH. Very beautiful, intellectually fascinating axis. And really, I was the first one, and with no data almost at the time. Maybe the PMC is in the skin, CRH is in the skin. So like almost 25 years ago, we formulated the concept that the skin has the HPA axis system in response to stress, and that it has adapted this from the central level because also you have this diverse stressors acting on. So this is organized the same way.
I see. So it sounds like what you're saying is, so classically, when we think about stress and stress hormones, we typically think about the hypothalamus and pituitary. There's a system set up in that part of the brain to release stress hormones, both within the brain and throughout the body, in order to prepare the animal to respond to stress. What you're saying is that there's actually kind of like a similar setup at the molecular and cellular level, actually in the skin, in terms of a stress response mechanism.
Exactly, and I will jump a little bit offside, but there is another stress response system, which is activation of adrenergic system, just release of the catecholamines. This is another system. So in the skin cells can also produce catecholamines. They not only released from the nerve ending, they are produced by the resident skin cells. But the production of catecholamines, it is by other people and we confirm this. So but let's come with the HPA axis. So the similar system is operating in the skin, and once we have more and more data, because we were also funded to study PMC in the skin and CRH receptors in the skin, so we suddenly started to realize that there is diversification. So this axis may not be as classical context dependent. It can be CRH receptor, PMC, cortisol, but depending on the stressors, it can be really so-called fragmented, and regulated in a different way. So in 2007, I have published a commentary in the Journal of Clinical Investigations under the title Nervous Breakdown in the Skin. I can send you this commentaries where really I have changed my mind. Just I come into conclusion that this is not the skin adapted from the central axis, but the central system has adapted this primordial responses to stress and perfected on central level.
So it sounds like you're saying from an evolutionary perspective, the sort of basic primordial stress response system that we see in the brain and the hypothalamus probably has as a precursor existed in the peripheral tissues like the skin of animals.
The skin or integument, because the central nervous system back far in evolution was not developed. So it was through evolution, everything was perfect. What we have right now is absolute perfection to the current environment. If someone believes to evolution, I believe. And so it is in the primordial stress response system has used the same molecules, the same receptors and signal transduction and shutting off mechanism.
And I see. So basically you're saying there's a very old system to respond to stress that involves the release of things like hormones, things like what we normally think of as neurotransmitters in the skin, and this can be found even in organisms that don't have a very well developed nervous system.
This is correct, and this is what has driven me in this commentary, and it was surprising that they really let me publish this, and even the JCI was preparing for me the schematic figures. I was by hand drawing, sending to them, and they were preparing. I said, oh, this is this, and finally, and I think it was perfect. So the idea was that this is because everything is in the skin, so the cells are close. So all of this interaction are very close, and this is somehow getting diffuse, like diffuse endocrinology or balkanization. I don't want to be politically incorrect. Balkanization, we know what does it mean, what was happening in the Balkans, but like balkanization of the certain orders, but everything has a logic, is context dependent or stochastic in nature. So this system, through the evolution, was, because it was almost perfect, and it was perfected and developed in the central HPA axis, which differed from the skin because there is anatomical dissociation. You have hypothalamus, pituitary, anatomically dissociated, so the signal can go very precise. And then you have another dissociation between pituitary and an adrenal gland, and then you have very important element which is in the HPA axis, and it has to be also it is not considered in classical way, but I think in the educated well it must be considered. It's the immune system, because immune system on the beginning was primordial, so everything was communicating. So the immune system also is part of this HPA axis because it communicates with the pituitary, it communicates with the adrenal glands, and it's getting shut off if you have this pro-inflammatory molecules produced by the HPA axis, the final reading of HPA axis cortisol, but also intermediates like ACTH is also acting as immunosuppressor. So all of these elements become separated, and then you have a perfect algorithm of our body. So I think this is very educational, and that skin. Another element is just the biogenic amines and the melatonin. So this melatoninergic system, but with the melatoninergic system, there is also a long evolution because on the beginning, the people predominantly thought that it is produced only in the pineal gland. When I was publishing papers on the first papers demonstrating that the melatonin can be produced, and we did very good biochemistry. It was really to provide the proof in the classical way, just when you look on precursor and product.
Yes. So it sounds like you're starting to say, so not only does the skin produce stress hormones and have sort of a stress system that is, in essence, a primordial version of the stress system in the brain, but it also produces things like melatonin. So we normally think of melatonin as being regulating our circadian rhythms and being secreted by the pineal gland of the brain. But this is, again, an example where the skin can also do something similar here.
Exactly. So with the melatonin, I was the first one to show the production, and I was facing the scrutiny because one of the directors of NIH, whom I know, I will not say what, so he was sending the emails to me. No, it must be artifacts. And I was sending back to him, no, this is not the artifact. We have a mass spectrometry data. So at the time, he never admitted, oh, I see that you want to study melatonin in the skin, but he never agreed with me at the time. But it's no question.
So light, is it dependent on light sensing in the skin, similar to the way that melatonin production centrally depends on light levels?
Probably light may play a role, but most likely is peripheral, which is independent on light. And it is really the production is probably for local purposes, because locally it will be monumental production. Mm-hmm. And the production of melatonin, once you start to analyze the property of melatonin, because other properties which are recognized and which were championed by Russ Reiter, is the antioxidative properties of melatonin by itself as a molecule, but also is melatonin is inducing the antioxidative protective mechanism and the DNA repair system. So once you start to put this together, it will be logical that the melatonin in the skin, the function is protection against the ultraviolet and sun, which is continuously damaging the skin. And you have production of the hormone, which is counteracting its effects. So
I see. So melatonin is an antioxidant, and it's not only involved in our circadian rhythms, which is what it's most famous for, but it's also involved in things like DNA repair. So in response to an environmental stressor like UV light, which can cause oxidative stress and DNA damage, melatonin is actually being used locally in the skin to help protect against that.
Yes, or to restore the homeostasis. In fact, there are really many areas to research. And one of the pure speculation which you can come out, okay, you have this stress UV during the summer, and now during the night, you have to restore, protect, to repair the mechanism, because otherwise you have the destruction of the organ. So intellectual, but we have to do more data, and the money for such research are not as available. And then during the resting period, it is repairing the DNA, pushing the cells which are not destined for reparation, because they may be precursors for the cancer cells, for apoptotic pathways. So through different mechanisms, and melatonin is doing this in the skin, which is, and I can also say the connection with the ultraviolet, that for example, we know the whole pathway for serotonin and then melatonin. It starts with the tryptophan hydroxylation. You will expose tryptophan, simple tryptophan, to ultraviolet radiation. You can generate 5-hydroxytryptophan. When if there is a decarboxylase, an amino acid, so it will be changed into serotonin. So there are also elements of this physical, not only enzymatic, physical chemical interaction of the light or of the short wavelength of the light. Then, with the melatonin by itself, is metabolized through the kynuric and indolic pathway. But let's say kynuric pathway, which is production of AFMK and AMK, which are also potent antioxidant. So these are different molecules which probably will act on different receptors, but also the metabolism or degradation of melatonin is activated also by the UVB, interestingly. To produce AFMK and AMK, you don't need to have, there is enzymatic pathway, but you don't need to have enzymes. Ultraviolet light will do this.
I see. So just the exposure to UV radiation can
to the environmental, to big environmental stressor can do this. So you have production of other molecules under the stressful damaging condition, which are also counteracting the biological effect because they induce the DNA repair mechanism.
Yeah, so there's a lot of built-in biochemistry here whereby the exposure to a stressor like UV radiation initiates the creation of molecules that serve protective functions, melatonin being one of them. I want to ask you a little bit more about the photobiology of the skin. So one of the things that I think is most famous in terms of how the skin responds to light is that we produce a lot of our vitamin D in response to UV radiation exposure. Can you tell us a little bit more about how does light induce vitamin D synthesis, and why does that happen? What is the vitamin D actually doing for the skin and the body?
I think this was I don't want to really put the speculation because the role of the UVB was established in the production of vitamin D3 for over 100 years. And 100 years ago, Tolum, I think the vitamin D production and discovered that it is, and the UVB is inducing production of vitamin D. There was a Nobel Prize for the production of vitamin D. And it was at the time in the 20th century, it was really important, rickets, and once you know, then you can cure many chronic and devastating in the outcome pathologies. So it is a simple photochemical reaction. 7-dehydrocholesterol, which is a final intermediate in production of cholesterol. So cholesterol, very important component of membranes and very important components of the body lipids, I think crucial, because cholesterol is also precursor for the metabolic transformation into precursor to pregnenolone, which is all steroids.
Yeah, it becomes, it's the precursor to all the steroid hormones.
This we vertebrates die. So this precursor is 7-dehydrocholesterol, and it is produced in the skin, is in other organs. And 7-dehydrocholesterol has the unsaturated B ring. This B ring is reduced by 7-delta reductase to produce cholesterol. In the skin, situation is a little bit different, in particular in the epidermis, because it can be, but if it is exposed to UVB during the summer, when UVB is in the spectrum of the solar radiation, the B ring is up, and this is I think it is physical-chemical beauty in biology. For me, it's one of the most beautiful photochemical reactions, which was adapted by all vertebrates living on the sun. So it is absorbed is absorbed this energy, and the B ring is broken and is changing the configuration, which is accelerated under the temperature, and it can go into the configuration of vitamin D3, which then is ejected and then is activated in the hydroxylation on the carbon 25 in the liver, but also locally.
Mm-hmm.
And
so
then,
UVB transforms this version of cholesterol into vitamin D3, and then
what
does the vitamin D3 do?
Vitamin D3 enters the circulation or is staying locally and is further enzymatically activated. And through two sequential hydroxylations in position 25 and then position 1, so it will be 1,25 dihydroxyvitamin D3, which has the fundamental role in the regulation of calcium metabolism in the body, bone formation, musculoskeletal system, but also regulate many other functions which are diverse. So this is one hormone. Of course, you can supply so the UVB because UVB it's not a big brainer. It's like Dr. Jekyll and Mr. Hyde. Depending on the context, is good. Vitamin D is bad. If you expose yourself too much, depending on your skin pigmentation, the skin cancer is guaranteed.
Mm-hmm. So UVB is bad because it can promote skin cancer if you get too much exposure, but it's also good because it's doing a number of other things that are beneficial.
Exactly. So with the vitamin D, and of course you can just prevent exposure from the UVB by oral supplementation. But oral supplementation of vitamin D, you go through GI, go through the liver, 25 hydroxylation in the liver, and it is like targeted most of this into this 125 classical pathway. I have discovered, and we have discovered together with Robert Taki from Australia, that there is alternative pathway, and the enzyme CYP11A1, which which transform cholesterol to pregnenolone, starting steroidogenesis, also recognize vitamin D3. And it's hydroxylating in different positions, producing several hydroxy metabolites. And these are also biologically active locally, perhaps.
Are you saying that so the vitamin D that's produced in the skin via exposure to UV radiation is a different version of vitamin D than what's produced in the liver?
It's the same.
Okay.
It's the same, but also it shows how accessibility and how our body can using the same precursor really make different balance. For example, if you take orally, majority will be hydroxylated in position 25. 25 hydroxyvitamin D3 is not recognized by CYP11A1. So once you hydroxylate in this position, so it is classical pathway. There is no other pathway for activation. But if you have unmodified vitamin D, so if it will bypass the liver, it's not hydroxylated or produced in the skin and injected into the circulation and go, for example, to the adrenal gland or in the skin, because CYP11A1 is also expressed in the skin and it is inducible by UVB, by the way. So everything context dependent. So it will activate vitamin D in different, producing the different hydroxy derivatives.
So the way that your body gets vitamin D dictates what it actually does with it. In other words, the vitamin D that you get through exposure to UVB radiation, if you walk outside in the summertime, is going to result in your body using the vitamin D in sort of different ways to some extent than if you were to take an oral supplement.
To some extent, because it will be from skin, it will be activated both. But this is not the end of the story. With 7-dehydrocholesterol and this photobiological transformation. because under prolonged exposure to UVB, so you have higher energy, more damaging, the configuration, because this photochemical is changing from D3 pre-D3 into tachysterol, which is really very, very difficult to study because it undergo easily auto-oxidation, so it's unstable. So we studied a little bit, very, very difficult. We come to conclusions, but we moved into lumisterol because there is another compound, lumisterol. So prolonged, and this is the most stable form, prolonged formation that this bearing is being resealed in different configuration, forming lumisterol. And before our discoveries, before 20 years back ago, everybody thought that lumisterol is inactive compound, that this is how the nature has formed if you have too much UVB. So the people are afraid also that if you use too much vitamin D orally, so you can produce hypercalcemia, you can even kill the person with the dosage. And never happens with the skin where this monumental concentration can be produced. So this is, and it was intellectually very stimulating, the hypothesis that, well, this is self-regulating system, UVB, more UVB, not vitamin D, but lumisterol, and this is like a sink protecting from the intoxication by vitamin D3. But let's say billions years of evolution, if something is formed, cannot be useless, because this is economy of the life. So it must be used for something. And what we have discovered is that the lumisterol is not the dead end product. It can be hydroxylated by CYP11A1 and by CYP27A1 to different hydroxy derivatives, which suddenly are biologically active. We tested this in the predominantly skin cells, and they act on the nuclear receptor. They do not act on the genomic side of VDR. They do not, but they act on the LXR receptor. They can act on the retinic orphan acid receptors as inverse agonists. They can act on other nuclear receptors and have the phenotypic effects. So, and which nobody was testing before we did. The lumisterol is circulating at high level in your body. So if enters the circulation, then it will become available for activation, either by CYP11A1 or by CYP27A1, which is expressed also in the liver. And about this, I will do what the consequences. So you have activation, and you have, I'm setting, and I'm not afraid to say, production of new types of hormones, because they will act hormones fulfilling the definition of the hormones through receptor-mediated mechanisms. So in this context, not only vitamin D3, on which we really very much, and everybody is focusing and is overlooking or is not knowledgeable on this, is lumisterol. And the lumisterol, similarly like a vitamin D3, would act as a prohormone. And I think that there are consequences, and it's a tip of the iceberg, because we started the study. And the question is always coming, like, you are vitamin D deficient, inefficient, or sufficient by measuring the levels of this hydroxy derivatives of vitamin D3. But you're forgetting, for example, about the lumisterol, which is also will be the consequence of high exposure to the UVB. And if it is activated, must have some biological activity, which we still don't know. It is like terra incognita.
So basically the total amount of UVB and other
it's a new hormone.
the total amount of UVB and other light that you're exposed to will dictate the specific pattern of different prohormones and therefore hormones that will be made in the skin and elsewhere, vitamin D3 just being one of them. But if you're exposed to high enough levels of UVB, you actually start to create this other prohormone molecule that will have different effects.
Exactly, you got it.
What about so everybody knows, well, I want to talk a little bit more about light sensitivity and photobiology of the skin. So obviously, UV radiation is famous for inducing mutations in DNA and stress in the skin. What about the skin's ability to detect other wavelengths of light? People are starting to talk more about red and near infrared wavelengths of light. How does the skin detect other wavelengths of light that potentially don't have damaging effects, but have other types of physiological effects?
Yes, there is. These are new areas which are developing, and unquestionably, it is accepted. For example, opsins are really are expressed in the skin cells, expressed in melanocytes or keratinocytes. And there are mechanisms of the detection, of the light detection, of the shorter, like blue light, or other further long wave, and they have the phenotypic effect. For example, blue light will have the effect on the melanocytes function, but this is still like a very. I'm not venturing into this. I realize, but because it is very difficult to investigate the field. There is effect. There are light receptors, unquestionable, but to nail down this, what is happening and how it is regulated, taking into consideration, because our focus will be the human skin. Let's focus. So there are different anatomical side of the skin, face, even on the face. Then you have the torso, you have the back, you have the hand, you have the feet, you have this different type of the skin, acral skin, without the adnexa. So they may have different mechanisms of the detection of external signals, and also the mechanism of the light detection. With the light detection is not easy to study, and for this, the good funding is required. Unfortunately, the funding is there is less and less funding by NIH and by or NSF, and the reviewers at how NIH always is saying, well, we have new hypotheses, and the best is NSF, because are the most really the good really review process. They look for new ideas and the new hypothesis to be tested. Unfortunately, National Science Foundation, if something is too close to the human, will say send to NIH. Once you go to NIH and something is too theoretical, you will be rejected, and there is no money to really just study and understand the mechanism.
So certain aspects of photobiology are hard to study, in part because the funding is hard to come by.
UVB also. I was fortunate why we were, because we could do the connection. It is not through vitamin D, but UVB acting on the skin, and we did in the murine system, can activate central, not skin, central. So brain sends the UVB through the pathway, hypothalamic axis. UVB could activate the propiomelanous signaling in arcuate nucleus. Then we had, and these mechanisms are different. UVB, and this is still unexplained, and we can hypothesize, but there is a need for more experience on this, can activate. We did the experiment with animals. You expose animals to the UVB, the skin, and then could shut off within 60 to 120 minutes, shutting off completely immune response in splenocytes, in the spleen cells. So spleen somehow detects the signal.
So somehow the body's detecting light.
It's going probably through neural transmission. So you're a neuroscientist, so too fast to go through hormonal must go.
Yeah. So within seconds, the body can detect light in the periphery and use neurons to communicate up to the brain, presumably, and then out to other organ systems faster than a hormone could get there.
And I must tell you, the spleen remember, because after 24 hours, because we have to set up in 48 hours, the immune responses of the splenocytes were muted. So it's not only you have rapid, but there is memory of this for a certain period of time. So these are fascinating areas because I think they could help in the therapy, very simple, very economical therapy without many drugs, biologicals which cost enormous amount of money, just good phototherapy, educated phototherapy, because we know what is the danger. We acknowledge skin cancer. In several neurodegenerative or autoimmune diseases like rheumatoid arthritis, like diabetes, like multiple sclerosis. And there are correlations, say, with the UV exposure to the UVB and just presentations of these diseases. So this could be simply applied in the educated way. And then I will have to complain how I'm funded by NIH. If on the other side, wherever I send the grant about the UVB, there is always this third reviewer. He wants to study the cancerogen as therapeutics. He must be crazy. So you will get the score, you will never be funded. Or they will say, well, study your vitamin D compounds and not the carcinogen. And this is it. But National Science Foundation, at least we went up to a certain level, was very open with it because it's a new biological. But National Science Foundation doesn't have sufficient amount of money.
One thing I want to ask you about is, so when people think about skin cancer, they think about, right, your sunlight exposure, your UV radiation exposure. What are the most common forms of skin cancer, and are they primarily caused by UV radiation exposure, or can skin cancers arise by other means?
Primarily, primary factor which is inducing skin cancer and the most frequent among any cancers are basal cell carcinoma. It's number one. Second is squamous cell carcinoma of the melanocytic origin, and undeniable clear-cut correlation between the UVB exposure. Why? Also, there is not need to be a rocket scientist, but DNA is a chromophore, is absorbed by UVB. And UVB induces the damage. And there is signature for the UVB damage, and this induces mutations. And also UVB generate immunosuppressive environments. Probably for good reason, immunosuppressive environment, because if you I will not speculate in this. And so everything will make a perfect condition for the formation of basal and squamous.
I see. So the UVB is not only able to induce mutagenesis, to mutate the DNA, which can lead to cancer, but it also suppresses the immune system, which might otherwise get rid of the cancer cell before it forms.
Yes, exactly. So everything in this place. So, for example, with vitamin D and melatonin, when we start to look at these molecules, so we see what this UVB is doing, and then let's say vitamin D3 and luminescence is a beautiful molecules because they only produced after UVB, nothing else. There is no biochemistry in the skin. So the nature has really adapted these molecules, first sense the danger, and the second counteract. So, and this is why, for example, vitamin D, active forms of the vitamin D have the photoprotective activities. They induce the antioxidative responses, and they really induce the DNA repair mechanism and inhibit cell proliferation. So yes, you induce the cancer, but there is these molecules after activation will counteract this process of carcinogenesis. And again, everything is concept dependent because different people have different not the capability, the propensity to develop the cancer, depending on the genotype. And there are different genes that you are more susceptible or not. And another which we see phenotypic, let's say, and this is a skin pigmentation, level of skin pigmentation. People with the light skin pigmentation without the facultative melanin production, which can be induced by UVB, skin cancer guaranteed. People with a darker skin, less probability of the skin cancer. Yes, they have the probability. You cannot, and it is unfortunately it's sometimes the pathologists ignore and can misdiagnose melanoma in the person with black skin, very black skin, because now he shouldn't get it. No, they also get it, but the probability is much, much lower. So
I see. So part of the function of skin pigment is actually to mitigate some of the stressful effects of UV light.
Yes, and with the humans, we have plenty of the example. How the gradient of skin pigmentation, you go towards the equator, black skin. And the big experiment performed by the British Empire by settling the Australia, the highest level of melanoma incidence in the world.
Yeah.
Where? In Australia.
Because you have light-skinned people that moved into an environment meant...
It's not only the light skin, because we have the, let's say about the pigmentation, you have Scandinavian, which have a lower level of pigmentation, and you have, for example, I can be politically incorrect because we speak science. We have the Irish people who have a lot of pheomelanin, this red pigment. And one of my friend, he's a chemist he was Italian chemist, and he was saying always to the Irish, we said, you black people, but you red people, you are not light people, you have too much pheomelanin. So the level of pheomelanin they have, it will be like African in Africa. But the pigment eumelanin is protecting. Pheomelanin is really bad news because after light or after different wavelength of UVR is producing this oxidative environment, producing the mutation. It is really one of the factors which really contribute to the melanoma genesis.
So, so you're saying that actually the pigment that fair-skinned, redheaded people have actually, in response to UV radiation, it's not only that you'd be in the presence of UV radiation, but that pigment can produce oxidative stress itself.
Right, pheomelanin. So to be better, lightly pigmented like some Scandinavians, than to have this redhead phenotype. And you think that, yes, the skin is white, but this is pheomelanin. This is a lot of melanin. So this person is not lightly pigmented, is not white. But
they have light skin, but they have a lot of pigment. It's just not a dark pigment.
Yes, it is a bad pigment. So for me, always it was really challenging through the evolution, the pheomelanin in the human population and this redhead phenotype should be eliminated because increased cancer. For some reason, not. So this is another interesting question to study. What is the use of pheomelanin, or at least some pathways connected to the pheomelanin, just which make you advantageous in
Yeah.
the evolution.
Yeah. It has this disadvantage of creating a oxidative environment in response to UV light, but it must be serving some advantage at a high latitude because it became common there.
Right. Yeah. I fully agree. You got it.
What about, so I want to talk about, so obviously the skin responds to light, as we've talked about. It responds to UV light. You can induce obviously mutations and cancer from too much exposure to UVB, but UV light also induces things like the production of antioxidants, vitamin D, the other things we talked about. I've also previously discussed on the podcast that UVB can liberate nitric oxide, which has cardiovascular benefits. Are there any other benefits to light exposure to the skin that tell us that we should get some sunlight, but just not too much?
I think that there is more than vitamin D and these molecules, because UVB as this potent stressor, and this is how we drove our hypothesis on the central HPA axis, can regulate homeostasis in a very regulated manner, like inducing sending the
full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.